Hello, welcome to Medical Area. In today's discussion, we shall talk about the pathophysiology of the H. pylori infection. As we are already familiar, the infection route for H. pylori is through the oral route. So we have our organisms getting down through the esophagus into the stomach. Now, I would like a kind one among you to go to the comment box and tell me the infectious dose of H. pylori. Now, tell me how many bacteria are needed for successful colonization of the gastric mucosa by H. pylori. Once H. pylori is in the stomach, there is a long chain of events that happens leading to the symptoms that the patient experiences. We are going to describe that by simply zooming out the stomach wall right this way. So in this illustration, I'm basically focused on the mucosa. But as we all know, beneath the mucosa, we have the submucosa, the muscularis externa, and the adventitia, which we're not going to be interested in this case. So in the mucosa, we have the epithelium, which in the gastric mucosa is basically a simple columnar epithelium. Then we have the basement membrane, the lamina propria, and the muscularis mucosae. So the three layers that we are going to be concerned with. Up here we have a layer of mucus. And in the stomach mucus is secreted by a type of cell known as the foveolar cell. Foveolar cells. These are found in the necks of the gastric pit around there, as opposed to the cells that produce mucus in the intestines, that is the goblet cells. In the stomach, we have the foveolar cells. So this is a layer of mucus. And then right here, I've tried to illustrate our H. pylori. It's a gram-negative rod. It is spiral-shaped, as I've tried to illustrate here. It has multiple flagellae on one pole. That is known as Lymphoid. So H. pylori has just landed in the stomach, and there is this harsh environment with a low pH, very acidic. So H. pylori has to figure out a way to survive in such an environment to escape the acid. It's got two mechanisms to do this. One of the mechanisms, very, very important, is the urease enzyme. And then the second mechanism is the acid inhibiting proteins, which I will talk about in a moment. So urease is an enzyme that is produced by H. pylori, and it breaks down urea to produce ammonia. Ammonia dissolves in aqueous solution to form a basic solution which neutralizes the acid, enabling H. pylori to survive. The second one is the production of the acid inhibiting proteins. These proteins go all the way and bind to the parietal cells in our gastric pit and inhibit the production of gastric acid. So that's where H. pylori is able to survive the acidic environment, but it cannot rely on these mechanisms all the way through, so it has to figure out a way to get to a safer environment. So it has to go through this layer of mucus and get beneath it an environment which is safe from acid. So one thing that H. pylori does is that it produces one enzyme which breaks down mucin. That enzyme is mucinase. This enzyme breaks down the mucus layer, and then our flagellae here start to beat to propel H. pylori all the way down through the mucus layer, and then it settles down here just next to our epithelial cells. Once it's down here, the environment is safe. It has got adhesion proteins expressed on its surface, with which it adheres to the epithelial cells. Once it's there, it causes damage through different ways. One of the ways it causes damage is by triggering an inflammatory reaction. And then secondly, which is actually very important, is that it produces two exotoxins. So one of them is the VAC A toxin. This is the vacuolizing cytotoxin A. And then the other one is the CagA cytotoxin. This is cytotoxicity associated gene A. So the protein is named so because it originates from the gene I've just mentioned. So the VAC A protein has a very undesirable action on the mucosal cells in that when it goes into the cells, it causes formation of vacuoles within the epithelial cell, and this results in apoptosis. Whereas the CagA protein is also introduced into the cell through a type of secretory system. It's a type 4 secretory system of H. pylori. It gets injected into the cell, and then this protein interferes with the integrity of the cytoskeleton of the cell. So here we have the microtubules, intermediate tubules, and so on. They are all interfered, and once they're interfered with, apoptosis also results from that and results in cell death. All these have a damaging effect on our epithelium. There are other ways in which H. pylori damages the epithelium. It produces other enzymes like the phospholipase enzyme, which actually breaks down our phospholipids in the membranes of the epithelial cells. Now, as this damage goes on, we have inflammation setting in, we have infiltrates forming, inflammatory cells migrating into the gastric wall, and then we have inflammation setting in. We have erosion of the epithelium, and that gives us gastritis. Then over time, as erosion goes on, so we have our epithelium getting eroded, and once it goes through the muscularis mucosae, we end up with a peptic ulcer. So there are other events that happen with chronic infection with H. pylori. Once this damage goes on over time, these cells have different responses to the injury. One of the responses is intestinal metaplasia. So what this simply means is that we have the gastric type epithelial cells changing into the intestinal type epithelial cells. And over time, as injury is sustained, it will actually progress to malignancy. There will be malignant change. So in this case, we are actually worried about the gastric adenocarcinoma. Gastric adenocarcinoma with chronic infection with H. pylori. The other malignancy that could arise from chronic infection with H. pylori is the mucosal associated lymphoid tissue B-cell lymphoma. This results from constant antigenic stimulation by H. pylori, which over time, as the B cells continue to proliferate in response to the antigenic stimulation, can result into the formation of a lymphoma. The other response to injury that these cells exhibit is, initially I mentioned that H. pylori is able to inhibit acid production in the gastric mucosa, but over time one of the responses to injury is increased activity. So in other words, we have increased secretion of hydrochloric acid by the parietal cells of the gastric pit. So this increased secretion will actually spill over into the duodenum. And in response to the injury, to the irritation, the duodenal cells also undergo a different kind of metaplasia. Instead, the intestinal type epithelium here changes into the gastric type epithelium. So we have gastric metaplasia happening here. Why is this so important? It's because initially H. pylori is not able to infect the intestinal type epithelium because the adhesion molecules it expresses on its surface are specific for the gastric type epithelium and not for the intestinal type epithelium. But then once this metaplasia happens, we have the epithelium of the duodenum, the first and second part especially, starting to look like the epithelium of the gastric mucosa, and therefore H. pylori can now bind to these cells and cause a similar kind of event that happened there, and that's how we end up with the duodenal ulcers, which are actually more common than the gastric ulcers themselves. Yeah, I think that is it for the pathophysiology of H. pylori. Thank you so much for coming with me till the end. In case you enjoyed this video, don't forget to hit the thumbs up button. And in case you wouldn't want to miss out on similar videos in the future, do subscribe to the channel and turn on your notifications. I would also be very happy to receive feedback from you in the form of comments, suggestions, questions, and I'll be sure to respond immediately. Thank you so much. See you next time.